So today I want to go over the virus, two viruses. The first one's ever sequenced and studied. One's called Phi-X174, and the other one's called Lambda, okay? Which both phages. I'll call them viruses. They infect E. coli only, which is a gut bacteria. They do not infect humans or mammals. Okay, we're safe from them. So I kind of, after getting over my pneumonia and COVID, I was put off doing anything to do with virus for ages, but it's been coming up for 2 years since I got over pneumonia, and I kind of suddenly feel keen to get back to doing viruses. They are a fascinating sort of beautiful kind of molecular entity, and it's a whole other world, and I think I've recovered from my virus infections enough to be able to talk about this again. So, okay, so Phi-X174, it was discovered by Robert Scheimer, okay, and it was used to first sequence DNA by Sanger in 1976, okay? First ever, it was because it was a relatively small DNA genome of 5,384 nucleotides long, okay? And it was the first ever, it actually led to the discovery of two genes completely contained in the larger gene. It also is evidence that because it's actually a single-stranded DNA, okay, which meant the base ratios they do not conform to rule where the concentration of adenine equals the concentration of thymine, and the concentration of guanine does not equal the concentration of cytosine, okay? This is what's meant by, this symbol means concentration in chemistry. So, because it's a single-stranded DNA, adenine, thymine, guanine, cytosine they don't join together in base pair, so you don't have to have equal numbers of them. So in this sequence of DNA, number of guanines doesn't equal the number of cytosines, the number of adenines doesn't equal the number of thymines, because they don't have to complement each other. So yeah, the basic map of the normally virus maps out like this. Yeah, you normally lay maps out like this to show where genes are. So you got the A gene there, these are just names, you can call them anything. B gene there, C gene there, D gene there. Within the G gene there's actually a section which is the E gene, okay? And then there's the J part there, there's the F gene there, G, H, okay? So these sections here are untranslated control sequences. And this these cases are examples where you have one gene contained entirely in another gene. So B and G is contained entirely gene. B and G is contained entirely within gene A. Gene E is contained entirely within gene D. But also, when Phyrex174 was discovered, this was a new discovery. Before then, it had been speculated, but nobody believed it. And then this section is unique because after it right adjacent to gene D and E, you got gene J, which is to do with ribosome initiation sequence. So there's a triple action in this section here. So this is the basic layout of the genes in Phyrex174. And all these genes specify different proteins. Okay. And then after, so things like structural proteins for the virus are various proteins that make the DNA get sequenced and expressed. But most of it will be capsid proteins and virus structural protein. And then Sanger then followed this with Lambda, which was then sequenced, which is a second virus studying is sequenced using the dideoxy method. And this was in 1977, okay? So this is a linear DNA. So I've been over Lambda. Just quick mention, Phyrex174. The structure of it is actually this spherical shape like this. It's not exactly spherical. It's more of a multiple-sided capsid spherical sort of structure, but it's sort of spherical thing with made of lots of proteins like this. Okay. It's sort of multiple-sided. It's lots of proteins make up the capsid coat. Okay, that's the shape of Phyrex174. This is completely filled in with proteins. So Lambda is, that's incorrect, it's linear DNA, 48,502 base pairs, okay? So it is considerably bigger than Phyrex174, but this is the second one ever studied. The genome is so it's got a 5' end. There's so you got 5' end, this sequence, and then DNA, and then the 3' end. Okay, that's more sequence, and then on the other strand you got 3' end, and then you got the same sequence, but then you got this sequence is basically repeated. Okay, so that complements that. So the CC GCC, for example, that goes GGC GG. So this can bind to that. These are called cohesive ends because they can actually, that can bind to that. So that should be actually 5', 3', 3', and it's a 5' end up here. They run out of space. So these can actually join together like that. So you get basically okay. So that complements that, and then you get, so the linear can actually fold up to become this circular DNA, okay? So And as it affects E. coli, there are quite a lot of genes in, which you can see from the image on this website. All the genes of lambda have been worked out. The way I believe this thing, I think, actually replicates by what's called rolling circle replication. It spins around. But this one replicates differently. This has got quite a complicated mechanism of the way it actually works. But the main genes are, so you've got C1. This is what's called a repressor. So it represses promoters. C2 and C3, these establish the lysogeny, which is to do with the way it infects cells. The CRO gene, which represses C1, and early genes. And then you got N and G, which is anti-terminators. Terminators stop the process of DNA replication or RNA synthesis. Anti-terminators stop the termination. So they stop, they basically stop. In this case it would be the creation of RNAs. They, I think, I haven't looked at this recently, but anti-terminators, I think, would actually stop if RNA is being synthesized, you get terminator, would stop it. Anti-terminators stop the termination. So, and then you get O and P genes and I genes and X genes. These are various other, creating probably proteins of various types, structural stuff. And then you get lots of other genes. So the overall virus has a structure like this. And it's made of there's lots of protein components making up the head like that, okay? So it's got a capsid structure like this, which is, it's meant to be, I didn't draw that very well, it should be more like that, but and then it's got a long neck that comes down like this, okay? So the protein structure spirals down, okay? And then at the end there's lots of legs like this that come off. And just to give you an idea of dimensions, that is approximately 300 Armstrongs, okay? When 0.5 Armstrongs, it's in one of my early videos, is the width, I think it's the radius of a hydrogen atom which is, and an Armstrong is equal to 10 to the minus 10 meter, okay? So, yeah, this is 300 of them. That's, I think hydrogen atom is about 0.5 Armstrong, one is 10 to the minus 10 to the meter. So they're pretty small. So that's a 10 billionth of a meter. So this is half of that. So that gives you roughly an idea of dimensions and what the thing looks like. So yeah, that's lambda. So this is the these two were the first ever DNA sequences of a genome to ever be, the first organism DNA sequences to ever be sequenced. First it was, sorry, that's lambda. First it was, FIREX-174, followed by lambda. Okay? This was chosen because it was a small length of DNA, which is a good one as a first ever attempt, and then this was another one that was worked on.